Welcome to Sticky Med, learning medicine, stick by stick.
Today we're doing a deep dive into a topic that really illustrates the incredible complexity and sometimes the heartbreaking fragility of our own internal security systems. We're talking about Ataxia Telangiectasia, or just AT.
Well, you have to understand that this is an autosomal recessive disease, so you need 2 copies of the defective gene. And it is progressive, it's relentless, and it leads to cerebellar degeneration, severe immunodeficiency, a huge cancer risk, and then those characteristic vascular lesions, the telangiectasias. But to get the whole picture, you really have to understand the job of the protein that fails.
Okay, well, let's unpack this then. Starting with the machinery itself, you mentioned the core problem is a defect in DNA repair. And I'm guessing that's what makes the cells of AT patients so sensitive to stress, like ionizing radiation, right?
Exactly. That drives this widespread genomic instability every single day.
So let's start with that molecular security chief then, the ATM gene. It stands for Ataxia Telangiectasia mutated. It's on chromosome 11, and it codes for this huge protein kinase, but its size isn't the point, its function is. It's the master regulator of the response to DNA double-strand breaks.
Right, and you have to think of DNA double-strand breaks as pretty much the worst thing that can happen to a cell's DNA.
A clean break.
A clean severing of both strands of the helix. It's like cutting the library's master catalog in half.
Yeah.
When that happens, the ATM protein isn't just the first responder, it's the general sounding the battle alarm. It detects the damage, it binds to it, and it kicks off this immediate complex signaling cascade.
I like that analogy, the general sounding the alarm. It implies there's this really swift, decisive action. So how does it get that message out to the rest of the cell?
It does it through a process called phosphorylation. A protein kinase like ATM doesn't just sit there, it acts like a switch. It attaches a phosphate group onto other proteins downstream, big names like P53, BRCA1, CHK2, and that essentially flips them from off to on.
So it's turning on all the emergency lights at once. And you mentioned this activation has 3 critical purposes.
3 jobs. First, it immediately establishes cell cycle arrest. I mean, if your DNA is severed, the absolute worst thing you can do is try to copy that broken blueprint.
Of course.
So ATM makes the cell just pause, buying precious time for the cleanup crew to get there.
And the second purpose is activating that cleanup crew, DNA repair. So it recruits all the machinery needed to patch up that double-strand break.
That's right, because if that break isn't fixed, it leads to massive genomic instability where, huge chunks of genetic material can be lost or rearranged. And the third purpose is, well, it's the cell's ultimate sacrifice. It's the safety net for the whole organism.
Apoptosis.
Apoptosis, or programmed cell death. If ATM decides the damage is just too great, too messy to fix, it triggers cell suicide. It removes the threat.
So in AT, that mutated ATM protein is non-functional. The general is asleep at the switch. The alarm never sounds.
Precisely. You have a profound failure. Cells with damaged DNA are just allowed to keep going through the cell cycle, proliferating these broken, unstable genomes. And this leads to 2 really tragic consequences. It promotes malignant transformation, and it leads to the death of very specific, highly sensitive cells in the brain.
Which brings us to the most defining clinical feature. That molecular failure sets the stage for the neurological impact, specifically ataxia. And this is usually the first red flag, right? It presents in early childhood, maybe age 1 to 4.
That's right. The ataxia shows up as this progressive difficulty with coordination, balance, and a very unsteady walking pattern, a gait disturbance that just gets worse over time. It's pretty recognizable, but the reason for it is where the real horror of this molecular failure lies.
And here's where it gets really interesting for me. Why the cerebellum? Why is the brain's coordination center so uniquely vulnerable to this specific defect?
It all comes down to the Purkinje cells. The root cause of the ataxia is the premature death of these highly specialized neurons in the cerebellum. And unlike most cells, Purkinje cells are postmitotic. They don't divide. They are incredibly long-lived and very, very metabolically active.
So wait, if they don't divide, why do they need the ATM protein so badly? I thought its main job was repairing damage before cell division.
That's a brilliant question, and it's a huge area of research. The thinking is that even non-dividing cells sustain this constant low-level DNA damage just from, living.
Oxidative stress, things like that.
Oxidative stress, transcription errors, all of it. In normal cells, ATM is always patrolling, fixing these little errors. But without a functional ATM protein, this background damage just accumulates and accumulates in these critical Purkinje cells until eventually it triggers their death. It's like a slow erosion.
Wow. That targeted cell death explains why the motor symptoms are so progressive and irreversible. But it's not just the gait issues, is it?
No, absolutely not. As the cerebellum degenerates, we see other hallmark signs. There's dysarthria, which is a specific slurred, slow, and very effortful speech pattern.
And one of the most distinctive visual cues is the ocular motor apraxia, that difficulty with voluntary eye movements.
Yes, the patients can't just smoothly move their eyes to a target. They often have to use this rapid compensatory head thrust just to get their gaze where they want it to go.
It's a physical workaround.
It's an involuntary strategy to overcome that deficit, and later on you often see involuntary movements like dystonia or choreothetosis.
That just paints a picture of this relentless deterioration. Let's shift now to the multi-system effects, starting with the immunodeficiency, because it seems a bit counterintuitive that a DNA repair defect would cripple the immune system.
It's a perfect example of how molecular biology determines systemic function. The immune system, our T and B cells, they need a kind of controlled chaos to work. To generate this huge arsenal of antibodies and receptors to fight every new threat, they undergo a process called VDJ recombination.
Can you break that down for us?
Sure. Think of the VDJ process like a molecular key maker. To create a unique key or receptor for a new virus, the T and B cells have to randomly shuffle and splice different genetic segments. And this cutting and pasting inherently involves generating and then immediately repairing DNA double-strand breaks. They have to break the DNA to rearrange it, then fix it instantly.
Okay, now I see the fatal flaw. If ATM isn't working, they can make the cuts.
But they can't repair them correctly. It leaves the DNA broken, and the immune cell is either non-functional or it just dies.
So the lymphocytes can't diversify properly.
Exactly right. And this leads to these profound deficiencies. Low counts of CD4 plus T cells, which hit cellular immunity, and a humoral deficiency with critically low levels of antibodies like IgA, IgE, and IgG2.
Which means these patients are not just getting minor colds. They are suffering from constant, very serious infections.
They are. The critical consequence is recurrent, frequent, and severe centopulmonary bacterial infections, lungs, and sinuses. And this isn't trivial. These chronic respiratory infections are a major cause of death, often leading to severe lung scarring called bronchiectasis. Life becomes a constant battle against common bacteria.
Let's touch on the last part of the disease's name. Telangiectasia. What are these?
Telangiectasias are small, benign clusters of visibly dilated capillaries. You can think of them as little spiderweb patterns of blood vessels. They're another sign of this underlying instability.
And their appearance is quite different from the neurological symptoms, timeline-wise.
Yes, very different. They appear later than the ataxia, usually after age 3 to 5, and they're a huge diagnostic clue because of their location. They're most prominent in the eyes, the ocular conjunctiva, and on exposed skin, like the ears and nose. Not life-threatening, but a very visible sign of that systemic failure.
Okay, let's move to what is maybe the most serious consequence of the ATM failure. Malignancy. This connects right back to that third safety protocol we talked about, the failure to trigger apoptosis.
It does. We said ATM's job is to kill irreparably damaged cells. When that system fails, those damaged cells are allowed to survive and proliferate, and the risk of cancer just skyrockets.
What kinds of cancer are we talking about here?
The predisposition is strongest for hematological malignancies, cancers of the blood. So we see a significantly increased risk of lymphomas and leukemias, specifically T-cell leukemias and B-cell lymphomas, which makes perfect sense, right?
Because those are the immune cells that were struggling with VDJ recombination in the first place.
Making them inherently prone to errors and genomic instability, yes.
And this heightened cancer risk, combined with the underlying defect, leads us to a really crucial, actionable point for anyone listening. The profound radiation sensitivity.
This cannot be overstated. Because of that fundamental inability to repair DNA double-strand breaks, AT patients are exquisitely, severely sensitive to ionizing radiation.
What does that mean in practical terms?
It means that standard doses of radiation from a CT scan, or certainly from chemotherapy, can be catastrophically toxic. A normal course of radiation treatment for cancer could cause severe, even fatal cellular damage throughout their body.
So it demands specialized treatment protocols.
Hello.
Okay, so given this whole complex picture, how is AT diagnosed and then managed?
The diagnostic path usually starts with clinical suspicion, that triad of ataxia, recurrent infections, and telangiectasias. Lab tests follow, and there's one very characteristic marker: significantly elevated serum levels of alpha-fetoprotein, or AFP.
And AFP's elevation is a really strong signature for AT.
It is. But the definitive confirmation is genetic, sequencing the ATM gene to find the mutation. And we also use a fascinating lab test called the chromosomal breakage test. Lymphocytes from the patient are cultured and exposed to X-rays. In AT, their chromosomes visibly shatter.
Wow, a visual confirmation of the defect.
It is.
So once it's confirmed, since there's no cure that restores ATM function, management must be focused on supportive care, right? Mitigating all these complications.
That's right. For the neurological side, it's intense physical and speech therapy to help maintain function for as long as possible.
And for the immunological failure.
The primary treatment there is regular immunoglobulin replacement therapy, IVI. It involves infusing concentrated IgG antibodies to supplement what the patient is missing. This really helps reduce the frequency and severity of those lung infections. It's essential.
And we have to circle back to that critical safety protocol, the radiation sensitivity.
Yes. Strict avoidance of diagnostic and therapeutic radiation is absolutely mandatory. It's a defining part of AT care. And because of that huge cancer risk, patients need vigilant, regular surveillance for those hematological malignancies, often starting very early in life.
So if we just synthesize this whole deep dive,
Yeah.
we've traced a single protein kinase, ATM, that fails in its job of guarding the genome. And that one failure cascades into cerebellar death, immune system collapse, vascular lesions, and cancer.
So what does this all mean? It means understanding that the ATM protein isn't just a gene, it is a fundamental guardian of biological stability. The disease really illustrates that life isn't just about building complex systems. It's about the silent, constant defense mechanisms required to maintain them.
And the incredible severity of Ataxia Telangiectasia really underscores how much we all rely on these silent defense mechanisms, like a single protein kinase, to ensure our fundamental integrity against damage we don't even realize is happening.
And we'd encourage you to maybe explore the vast interplay between all the different DNA repair pathways and our overall health. When those pathways fail, even systems that seem totally unrelated can just collapse.